In the last mini lecture, we talked about monohybrid crosses. We talked about how you could predict the offspring in a cross between two parents, each of which having looking at one character with two distinct traits. Moving forward into this mini lecture, we're going to talk about two parents, each with two characters and a total of 4 possible traits, 2 traits per character, and seeing what the resulting outcome is going to be. So, a little bit more complicated than what we just did, but not terribly much when you realize that dihybrid crosses are really just 2 monohybrid crosses that you multiply the results for. So we are going to conduct a 2-factor dihybrid cross. It's just like doing a monohybrid cross. You just understand the gametes form independently of each other. There's independent assortment and the independent segregation of alleles. If you take a look at this ear of corn, you'll see that there's 2 characters with a total of 4 traits. The characters here are seed color and seed shape. The different traits are purple or white, and the other one for seed color and for seed shape, you've got sweet, which are the sort of wrinkly ones, and flint, the big fat ones. So 2 characters, seed color, seed shape, 4 traits, purple, white, sweet, flint. Let's see how it all works out. First thing we need to do when we're looking at this more advanced work is figuring out how to make gametes, because it's not just take one thing and one cell and split it in 2. You've got to figure out all the possible combinations of gametes. We're going to start off looking at the most complex, the heterozygous, fully heterozygous. So in this case, I've just chosen the random letters R, the random alleles R and Y. We have a big R, big Y, little y. To make gametes for it, you can use what's known as the FOIL method from algebra. Remember that? First, outer, inner, last. So you take the first 2 alleles, big R, big Y, the outer 2 alleles, big R, little y, the inner 2 alleles, little r, big Y, and the last 2 alleles, little r, little y. And there are all of your gametes. So that's one way of doing it that works well for dihybrid crosses. Another method I use is sort of what I call the branched line method. If I've got alleles big A, big B, little b, I have 2 possibilities for A. It's either going to be big A or it's going to be little a. So I put them right above each other. You can start with a single point. 2 lines come off of it. Because we are diploid, we have 2 possible options. Then, off of that A, I'm going to put 2 more lines because I've got 2 more options. Big B and little b. After that, I just follow the lines. So one of my gametes is going to be big A from the first position to the very top B, so I get AB. Then I go underneath it, big A, little b, so I get A, big A, little b, then little a, big B, then little a, little b. This branch line method works really well, especially when you're doing not just dihybrid crosses, but say trihybrid crosses or pentahybrid crosses or octahybrid crosses, when you're trying to coordinate many, many, many different alleles. So these are all the possible combinations from that particular organism. Now, you might have a, big B, little b, or something like that. If that's the case, you only have one possible option. You've got a big A, and then big B, little b. So AB, big B, big A, little b. You wouldn't have the little a if it was just big A, big B, little b as the genotype. So 2 possible ways. Choose whichever way you feel most comfortable with. We would then combine different organisms, just like you would do a normal Punnett square, except this time instead of being 4 squares, it's 16 squares. There are a lot more possibilities. This particular image shows what would happen if you combined big R, big Y, little y, two big R, big Y, little y. So two fully heterozygous organisms mate. We took the gametes that we made 2 slides ago, we put them onto this Punnett square. One of the parents is at the top, one of the parents is down the side. And you'll notice that in a dihybrid cross, each gamete has 2 different alleles. If you've got 2 of the same alleles in a gamete, any 2 R's or 2 Y's, you're doing something wrong. That's a big red flag. In a dihybrid cross, you should always have 2 alleles in your gamete. Any more, any less, and something's wrong. Double check your work. Then it's just the exact same as before. You're going to take one gamete and fuse it with another in order to get a genotype that you can interpret. So for instance, the top left corner has big R, little y crossed to, little r, little y crossed to little r, little y. You cross them together and you see that you get a, both are homozygous recessive. So it's going to be, in this case, fully round and yellow seed pods. To interpret these, typically you're going to just count up the phenotypes. You're not going to look as often at the genotypes. When you cross 2 heterozygotes, you will typically, in a dihybrid cross, fully heterozygous, you will end up with a 9 to 3 to 3 to 1 ratio. 9 display one dominant trait, 9 display both dominant traits. 3 of them will display one dominant trait and one recessive trait. 3 of them will display the other dominant trait and the other recessive trait, and 1 of them will display both recessive traits. So, lot going on there in order to interpret these. There are 16, so there's a lot of potential for making a mistake. You have to count carefully. You have to be precise in how you write out your alleles, because if you write too quickly, you may misinterpret them. Take your time, do it right. Be able to predict the next generation. So we're going to do an example of this. We're going to cross a pea plant that's heterozygous for purple flowers and homozygous dominant for yellow seeds, with a plant that's heterozygous for purple flowers and homozygous recessive for green seeds. Give the expected phenotypic ratios of the offspring. Just like last time, take it a little bit at a time, just one sentence at a time, and start with defining your alleles. The first alleles we got are big P is purple, little p is white. Big Y is yellow, little y is green. From there, you're going to, now that we've got our alleles labeled, you got to figure out the genotypes of the parents. Our first parent is heterozygous for purple. Hetero means different, so it's going to be big P, little p, and homozygous dominant for yellow. All right, it's homozygous and dominant, so big Y, big Y. So that's one of our parents. Little p, big P, little p, big Y, big Y. You're crossing it with a plant that is heterozygous for purple. Again, hetero means different, so big P, little p. And homozygous recessive for green. Little y, little y. So now we know our parent genotypes. From our parent genotypes, we can make our gametes. You can use either the FOIL method or the branched line method. What you'll find is that the first parent has a big P, and the only other option is big Y. So it's a big P and a big Y. And the other gamete is going to be a little p and a big Y. Now, could you make all 4 gametes for this? Yes. You could put first ones, big P, big Y. Outer ones, big P, big Y. Inner ones, little p, little big Y. Outer one, last ones, little p, big Y. You have 4, but you're going to find that because 2 of them are exactly the same, you're just getting redundant answers. You're just doing extra work that you don't need to do. The same with the other parent. You've got big P, little y, little y. You've got a 50-50 chance of getting a big P or a little p, but a 100% chance of getting a little y. So the only 2 possible gametes are big P, little y, and little p, little y. You take those gametes. That's the hard part. Once you got the gametes, it's all down the hill from there. You put them into your Punnett square. It could be 16 if you went all the way out with 4 each, but because we shortcutted it and realized that we would just be putting in redundant gametes, we've only got a 4 square Punnett square. You got big P, big Y at the top, little p, little y next to it, and then big P, little y going down the side. One parent at the top, one parent going down the side. Then you do your fertilization event to give you offspring with big P, big Y, little y, and so on. Okay, we've got our values now. We've got our F1 generations genotypes. We need to give the expected phenotypic ratio. So how many of these will have purple flowers? 3 have purple flowers. 1 has white flowers. Of those, how many are both purple and yellow? 1, 2, 3 are purple and yellow. That is, they have a big P and a big Y. And only one of them has a little p and a little y. Well, 2 little p's and 2 little y's. So our genotypic ratio, nobody cares. It wasn't asked for. Our phenotypic ratio is 3 to 1. 3 of these organisms have purple flowers and yellow pods. 1 of them has white flowers and green pods. Let's do another. We're going to cross a plant that's heterozygous tall and homozygous for green seeds with a plant that's short and also homozygous for green seeds. So we've got the same basic relationships. I'll tell you, tall is dominant to short, yellow is dominant to green. What are the phenotypic ratios here? Okay, first step, write down your alleles. Big T is tall, little t is short. Big G is yellow, little g is green. So far, so good. You can use any alleles that you want. These are the ones that I chose, but it's all, choose what you wish. All right, next step, figure out what are the parents. We have a plant that is heterozygous tall. Hetero means different, so big T, little t. And homozygous for green, little g, little g. That plant is crossed to one that is short. Now, in order to be short, you cannot have a big T allele. That means that it must be homozygous recessive, little t, little t. And also homozygous for green pods, so little g, little g. So we got 2 parents, one tall that's a heterozygote, one short that's homozygous recessive for both traits. Just looking at this, we know for sure that every single one of these offspring will be pure breeding green because there are no possible alleles to make them yellow. They just do not exist. So this is a much simpler problem than we've gone through before. Coming up with the gametes, we've got our the top parent gamete is a big T and a little g, and a little t and a little g. Big T, little g, little t, little g. Those are the only possible gametes. Could you go all 4 out? Yes, you could. Do you need to? No, because it's redundant. Even a more extreme example of that is the bottom one. The only between little t and little t, the only possible choice you get is little t. The little g, the only choice you get is little g. So there's only one possible gamete that will produce. Little t, little g. You put them into a Punnett square, and you find quite easily you've got one that is big T, little g, little g, and one possibility that is little t, little t, little g, little g. So figure out your ratios. Genotypic ratio is not asked for. Phenotypic ratio is 1 to 1. A tall green to a short green. Moving a little bit farther beyond the dihybrid cross, you may get a trihybrid cross. A trihybrid cross works with 3 possible characters, each with 2 traits. So you'll end up with 8 different gametes for each one, or 8 potential gametes for each. That makes instead of a 16 square Punnett square, a 64 square Punnett square. So you're getting a little bit bigger. Is it possible to do them using Punnett squares? Yes. It takes a very, very long time. And then imagine doing a tetrahybrid cross that has 256 squares, or a pentahybrid cross with, I think, 1028. You want to move away from using Punnett squares, just like you moved away from using number lines in elementary school. And instead, you could use simple mathematics to describe it. You could multiply the frequencies that you get for the monohybrid crosses, the individual monohybrid crosses, to each other to give you a combined statistical value for each potential genotype you're looking for. Am I going to go into it right here? No, I'm not. If you were in class, I'd do a lot of writing on the board. For this course, I am not going to have you do trihybrid crosses. I'm just having you stop at dihybrid crosses. Will you have to do dihybrid crosses on assessments and things like that in the future? I'm sure you will. Know how to do it, not just for these simple Mendelian dominant recessive traits, but also when we look forward into sex linkage or codominance or incomplete dominance, terms you haven't heard yet, so don't freak out, but you're going to be learning about them. You could do dihybrid crosses using all of these Mendelian characters. Just to give you a bit of extra practice here, so there are some questions. Work through them. See if you can figure it out. If you can figure these out, you should be good to go. So conducting a dihybrid cross is just like conducting a monohybrid cross, except instead of dealing with one character and two traits, you're dealing with two characters and four traits. The hardest part is determining your gametes. And to determine the gametes in a dihybrid cross, you could use the branched line method, which is what I prefer, or you could use the first, outer, inner, last method. Whenever you cross two heterozygotes, fully heterozygous dihybrids, you're going to see this amazing ratio of 9 to 3 to 3 to 1 every single time. And when you're writing your phenotypic values, typically you're going to write two dominant traits, one dominant, one recessive, the other dominant, the other recessive, and then the both recessives. When you cross two fully heterozygotes, you tend to see 9 to 3 to 3 to 1 in those ratios. These content review questions are here to focus your studies. The next mini lecture is going to be about how to read a pedigree, how to read a family tree, and use that family tree to determine genetic information, to figure out how a condition is passed on from one generation to the next. Is it going to be autosomal dominant? Is it on the all the chromosomes except for the X and Y chromosome? Is it the sex-linked condition? Is it any number of things? Is it dominant? Is it recessive? What are the possibilities for the next generation? So in the next mini lecture, we'll figure out how to read a family tree.